Among several existing protocols for tissue preparation for transmission electron microscopy, we selected methods that yield optimal structural preservation and high-resolution micrographs to perform morphometric analysis of synaptic vesicles distribution at presynaptic terminals. The high-contrast images obtained with these methods allow to quantify parameters such as distance of synaptic vesicles from the presynaptic membrane, number of vesicles docked at the presynaptic membrane, and intervesicle distances. All of these parameters are indicative of synaptic function. Since alterations in the spatial organization of synaptic vesicles may be associated with the disruption of synaptic function, these methods have been used to examine the effects of general anesthetics on synaptic development. These methods could also be used to examine the effects of any drug or treatment on the detailed morphology of synapses to provide insight into their function.
Begin by preparing osmium tetroxide for post-fixation of rat brain sections previously fixed with paraformaldehyde and glutaraldehyde via vascular perfusion.
Sections become rigid after treatment with osmium tetroxide. Therefore, tissue handling requires attention from then on. Also, before adding osmium, make sure that your sections are flattened, as any fold